Thanks for joining me. You'll notice new shirt. This is the quality construction that you only get when purchasing from Eddie Bauer. So be sure to check out their shop, see what they've got available, and you won't be disappointed. Trust me. What we were talking about last time is we were talking about DNA. We talked about alleles and different versions of traits and different versions of DNA that you can inherit. And what I mentioned is we can actually predict the traits that some organism is going to have based on its parents. And that's what we're going to learn about today, is something that's called a Punnett square and how we can use that to help us predict the traits of offspring. And we're going to do this by working through an example. So here's our example. In peas, we're going to say that peas have purple flowers, and that's a dominant trait over white flowers. What that means is that purple is dominant while white has to be recessive. It says if there are two heterozygous peas and they are bred together, what are the chances that the offspring of those peas are going to produce white flowers? So this is kind of a standard question that you would end up seeing when you're dealing with Punnett squares. You will always end up getting information, usually about a couple of parents. You'll be able to determine their genotypes, and then you can use that to make your predictions. To do this, there's pretty much some clear-cut steps that you can follow in your problem solving, and that's what I wanted to walk you through. So the very first step is to determine the genotypes of the parents. And so if we read through this question says that we have two heterozygous peas. Now remember, the genotype is the letters that we get. It's the actual genes that are carried by that organism. And so we're going to have P number 1, and we need to decide what letters it actually gets. Hetero means different. Zygous, remember, means pair. So this has a different pair of alleles. And I'm just going to use F to talk about flower color as my letter. So the way that I would write this is one capital, one lowercase, to show that it has a heterozygous trait. Now, there is a second P, P number 2, which is also heterozygous. And so those are our parents' genotypes. We need to know our parents' genotypes because they're going to pass their DNA down to their offspring during reproduction. And so their genes, we need to know those in order to actually figure out what those kids can actually have. So our next step is to draw a Punnett square. So a Punnett square is literally just a big square. So what we're going to do, leave a little bit of room to write at the top and room to write on this side. Make a large square. Now this square we're going to cut in half horizontally, cut in half vertically. And that is what a Punnett square looks like. The reason we make this is for our next step, which is to split the parent alleles and place them on the top and the side of the Punnett square. So what that means is parent number 1 has two alleles, a capital F and a lowercase f. And I am going to write these on the top of my Punnett square, but you notice I actually split them apart. The reason I split them apart is because during reproduction, those alleles actually do get split into separate cells, and only one of them gets passed down to an offspring. And so you end up splitting those now. And when we start talking about meiosis, keep this in mind because it's actually has a lot to do with what's happening there. Since we did that with one parent, we're also going to do it with the other parent. We're going to split this parent this parent's genotype up, and we're going to write one letter here and one letter here. The biggest mistake people make with Punnett squares is they don't split this apart, And so they'd write like capital F, lowercase f, capital F, lowercase f, capital F, lowercase f, and same here. And then in the center, you end up having 4 letters. And since we're dealing with a trait that only gets 2 letters, you can't inherit 4 letters, right? So our next step is to complete the square. And so what we do there is we take the letter from the top and put it in the boxes below. So capital F here, capital F there. Lowercase f comes down into this box, lowercase f into this box. These ones we put across. So capital F goes here, capital F goes here, lowercase f goes here, lowercase f goes here. That's how we complete the square. So now what we need to think about is, what does this stuff actually mean? So what this means is these are the possible offspring that they could have as parents. When you have offspring, it's like rolling the dice. You don't get to say, okay, this is going to be kid number 1, this is going to be kid number 2, this will be kid number 3 and 4. It's totally up to chance. It's all about probabilities. And so each of these 4 options that you see in the box, each of those is one chance of what the child could end up having. And so let's think for a minute, what are the actual genotypes of the children? Well, the genotypes are the letters. So if you're a child, you could end up being homozygous dominant, 2 capital F's. You could end up being heterozygous, capital and lowercase. Or you could be homozygous recessive and inherit 2 lowercase. It's also possible that you could, these are really the only possibilities for genotype. The other thing we need to think about is, what are the phenotypes? Remember, phenotype is the actual physical type, the traits that the organism is actually going to show. In this case, the trait we're talking about is flower color. And there are 2 possibilities that they could get. They could get the dominant trait. If you get the dominant trait, you're going to have purple flowers. If you get the recessive trait, you're going to have white flowers. And here's why we did this, is because we need to answer the question, which is, what are the chances that the offspring will have white flowers? So what we can do is figure out the ratios of everything. Remember, this is out of 4. So there is a 1 out of 4 chance, or a 25% chance, that they will be homozygous dominant. Same thing, a 1 out of 4 chance that they're going to be homozygous recessive. A 2 out of 4, or a 1 out of 2, however you want to say that, but 50% chance of being heterozygous. Now, their chances of inheriting purple flowers, though, is 3 out of 4. There's a 3 out of 4, or 75% chance that they are going to inherit purple flowers. There is a 1 out of 4 chance that they're going to inherit white flowers. So the actual answer here is that there is a 25% chance that the offspring will inherit white flowers. Now, when we think about probability, it's probably important to think about, what did we actually figure out. Remember, there's a couple of kinds of probability. There is what is called the theoretical probability, the theoretical chance of that happening, and there is the experimental probability, what you actually find out if you were to do this test. So a great way to think about it is a coin, right? A coin has an a theoretical probability of 50%. There's a 50% chance that when you flip a coin, it's going to come up heads or tails. But if you flipped a coin 10 times, that doesn't mean exactly 5 of them are guaranteed to be heads, while the other 5 are guaranteed to be tails. That the actual testing of it is the experimental probability. And so that is much, much different. If you're dealing with experimental probability, that's when you're actually measuring it. So a great thing to think about, if we go back to what we saw under the camera, let's say you bred these peas and you somehow got, let's say, 100 pea plants from them that you could grow. How many would you expect to be purple? Well, you should expect 75% of them to be purple. So since there's 100, expect 75. In reality, though, probably not going to be 75. It should be very close. And that ratio of 75 to 25, like the 3-to-1 ratio, should pretty much hold up. But you don't really know because it's an experimental probability. You have to do this like over and over and over again to make sure you confirm. But that's an important thing to keep in mind when we deal with probabilities. And so that's how we carry out a Punnett square. It's actually not super difficult, and it's a lot of people really enjoy these. And so I thought our next lesson I'll throw a little twist in here so we can see a different style of genetics problem as we work forward. Okay, so thanks for watching. If you have any questions, make sure you throw those at me so that I can help you out, and have a good day.